They're all GABAergic, and they show increases in excitability in the context of injury. So our proposed model was that activity of these GABAergic neurons is at least one of the sources for the inhibition that is seen in the zona incerta. We validated this connection, this anatomical circuit, by using retrograde tracers. So we injected a fluorescently labeled retrograde tracer in the zona incerta of PKC delta reporter animals. And we injected this in the zona incerta. And then when we look at the central amygdala, we can see that there's very nice colocalization of PKC delta neurons that picked up the tracer from the zona incerta, validating that what looked like terminals in our anatomical studies was indeed a connection. We further looked electrophysiologically. So using optogenetically assisted circuit mapping, we injected channel rhodopsin in the central amygdala, and then we made acute slices of the zona incerta and patched the neurons from the zona incerta in response to light stimulation of the terminals that were coming from the CEA. And as you can see in these representative traces, we see these quite robust inhibitory postsynaptic potential, demonstrating that, again, this is a functional synapse. And more importantly, in the context of injury with cough animals, we see decreases in pair pulse ratio, which is consistent with higher presynaptic efficacy in this pathway in the context of injury. So in order to now bring it back to the animal and the behavior, we performed behavioral experiments similar to what we did in the CEA, but now it's in the zona incerta. And I'm focusing only on the pinching experiments. But what we see here is that when we inhibit, and this is the simplified model, when we inhibit the zona incerta neurons, consistent with our model and with Asaf's lab data, we can create hypersensitivity that resembles injury but occurs without injury in control animals. And then when we activate the neurons in the zona incerta, presumably blocking or reversing the inhibition from the CEA, we can reverse the cough-induced hypersensitivity that we observe in the nerve-injured animals. So we feel that based on these findings, we can add to our model that one of the circuits that is mediating this cough-induced hypersensitivity is via outputs from the PKC delta neurons to the zona incerta. Then I'll spend the last few minutes telling you about the experiments that we've done evaluating the inputs that are coming from the CEA. And we're really excited about this project because we decided to start with the classical pathway that has been described for so many years. The main input from the main nociceptive inputs from the CEA have been described as coming from the parabrachial nucleus, the spinophotoamygdaloid pathway. This was characterized in the 80s and 90s with electrophysiology, demonstrating that the neurons in the CEA that receive inputs from the pons respond to noxious stimuli. And also anatomically, they've been validated, they've demonstrated these inputs that go all the way from the periphery all the way to the central amygdala. Now, the reason I'm excited about this project is because recent work that have used similar approaches to what I showed, either chemogenetic or ablation of neurons that go from the parabrachial neurons to the central amygdala, they've shown no effect in the modulation of behavioral responses to noxious stimuli when this pathway is modulated, whether it is ablated or if it's chemogenetically activated or inhibited. So these results were puzzling based on all the previous work. Then in addition to that, recent work has shown that consistent with anatomical studies, again from the 90s, that the spinal inputs that come to the parabrachial nucleus, they don't target the CA projecting neurons. Instead, they target thalamic neurons. In addition to that, this beautiful work from Sarah Ross's lab demonstrated that the neurons that receive inputs from the parabrachial, the CA neurons that receive inputs from the parabrachial neuron nucleus are involved in the affective aversive components of pain, but not the somatosensory components of pain. So why is this all puzzling? Well, people like me that have studied this pathway for over 2 decades were very surprised to see no modulation of somatosensory component, because we know that this pathway undergoes injury-induced plasticity. Neugebauer lab has shown that in the context of injury, in vivo and also in acute brain slices, there's potentiation of synaptic transmission in that pathway, and the neurons in the CA that receive inputs from the pons are sensitized in all the classical Clifford-Wolff definition of sensitization happens in the central amygdala in the context of injury. So this is all correlational, so it could be that this is what's mediating the affective component that Sarah Ross's lab demonstrated. But what we hypothesized was that perhaps this pathway is involved in driving hypersensitivity in the context of injury, not necessarily at baseline, which is consistent with the body of work that all of us have been producing in the CEA, demonstrating that the CEA is not involved in driving the baseline, yes, no, do I feel pain? But instead, what it does is that in the context of injury or in the context of stress, it functions as a rheostat that drives pain up or down. So we, of course, first wanted to validate that the work that Volker and many others have done over the years stands in our hands, and we used optogenetic assisted circuit mapping, so similar to the experiments that I showed before, but now injecting channel rhodopsin in the parabrachial nucleus and then recording the responses, the optically evoked responses from the central amygdala neurons. And what we see is that both somatostatin and PKC delta neurons receive strong glutamatergic inputs from the parabrachial nucleus. These responses are monosynaptic and are independent of the firing type of the neurons. We further demonstrate that if we use CFOS monitoring to look at the recruitment of the CA projecting PV neurons in the context of noxious stimulation, when we look at just pinch-induced CFOS, we see consistent with other studies, including Sarah Ross's, that the number of CFOS positive neurons in the parabrachial neurons increases with pinch. But importantly, when we look specifically at the neurons that are projecting to the CEA, which is basically what how we did this is we injected a retrograde tracer that expresses CRE in the CEA, and then we injected a CRE-dependent M-cherry in the parabrachial, so that all the neurons in the PV that project to the CEA will pick up the tracer that expresses CRE, and then they'll turn red because they have M-cherry. And what we're seeing here is that there's increases in the pinch-induced CFOS in the PV neurons that project to the CEA, suggesting again or confirming that the PV neurons are indeed undergoing plasticity in the context of injury. So in order to look at the behavioral modulation, now we used a similar approach, so an intersectional approach where we inject the Cre-expressing retrograde tracer in the CEA, and then now instead of just the fluorophore, we inject the parabrachial with a Cre-dependent inhibitory DRED. So now when we put CNO, the neurons that go from the parabrachial to the CEA will be inhibited. And when we do these experiments and at baseline, so no injury, what we see is that consistent with the work that I showed before from other labs, there's absolutely no effect of modulating these neurons. So you can completely get rid of these inputs, and the what I call the ouch component will still be there. The animals will still detect the acute nociceptive processing. Now, when we do these experiments in the context of injury, what we see is that now the neurons, the animals that have inhibitor, that the CA projecting parabrachial neurons are inhibited, now in all 4 modalities, they have reversal of the hypersensitivity in the context of injury, and only in the treated paw, with no effect on the ipsilateral paw in any of the modalities. So this was really exciting and a big relief, because if not, then it questioned the pathway that we have been studying for so many years. So we're really happy to see this. And I felt that for this particular crowd is important because it highlights it's extremely important to evaluate that transition between acute to chronic pain states, and also to acknowledge that and be careful in the interpretation and conclusions that we make in studies that are evaluating acute nociception, and how we then can make overall assumptions that are related to persistent pathological pain, which clearly shows that, all of us have been showing that that's not the case. And here is a very clear example of that. The last piece of data that I want to show is something that we're also quite excited about, and it's a brand new data that we're going to start following up to figure out what's going on. But basically what we have shown is that when we do these experiments, the same experiments, so modulating the CA projecting PV neurons, if we then repeat the test 3 weeks after the injury, now there's no contribution of these neurons to hypersensitivity. So it seems to indicate that the contribution of the PV to CA pathway to injury-induced hypersensitivity is time-dependent and occurs only at the beginning of the process. It's like with the learning and memory similarity would be like in the initiation and not necessarily the maintenance of the hypersensitivity. And I don't feel comfortable showing the data from the CEA, but the exciting part is that with our not systematic observations in the CEA, all the modulation that we have done in the CEA, we see it for any time after the injury. So demonstrating that or suggesting that while the inputs from the PV are involved in getting the hypersensitivity established, it looks like the CEA and other brain mechanisms via descending modulation are the ones mediating the maintenance or the long-lasting hypersensitivity that we observe after